Hello, my name is Breanna Pence and I conduct research at the University of Texas at El Paso under the mentorship of Siddhartha Das through the Research Initiative for Scientific Enhancement or RISE program. I currently work with the intestinal protozoan parasite Giardia lamblia, and I'm currently working to determine if variant surface protein and exosomal vesicles may be linked in parasite's virulence. I have worked in the lab for 3 years and have learned many techniques, and I wish to pursue graduate school. Giardia lamblia is responsible for the transmission of giardiasis worldwide. Symptoms include diarrhea, abdominal pain, weight loss, and malabsorption. It is commonly spread by the fecal-oral route via contaminated water. The cyst is the form of the parasite that is ingested, then trophozoites are released from cysts, and then they colonize the small intestine, causing symptoms. Routine culture of Giardia in vitro involves growth in an anaerobic environment. Glass tubes filled with TYDK medium is the most effective way to achieve this environment. To start an experiment, trophozoites are cultured in flasks until confluency in order to obtain a higher amount of cells. Shown is a monolayer of trophozoites at 100% confluency at 40x magnification. The project involves determining if virulence is linked between the parasite's membrane lipid rafts, variant surface proteins, or exosomal vesicles. Lipid rafts are cholesterol-rich and sphingolipid-rich areas of the plasma membrane specialized in cell signaling, membrane fluidity, and protein trafficking. Previous lab work has shown that lipid raft inhibitors hinder the production of viable cysts in Giardia. The control cells have thick and apparent cyst walls stained in red, but the nystatin and oseltamivir treatments show cells with less fully formed cyst walls. Further, stained in green is a particular trophozoite protein that is only visible in the treatment cells, which shows that these cysts still have trophozoite features and would not be viable. Variant surface proteins, or VSPs, exist on the surface of the trophozoite. The parasite can switch between VSPs throughout its life cycle, allowing the parasite to evade the host's immunity. My current goal is to determine if virulence factors such as VSPs are released or produced by Giardia via microvesicles or exosomal vesicles through lipid rafts. This is being done via confocal analysis, nanocyte analysis, and mass spectrometry. Confocal analysis involves staining particular proteins or structures of the parasite using a fluorescent antibody. The sample is then mounted onto a microscope slide for imaging. This image is a stain of the trophozoite's lipid rafts using a fluorescent green choleratoxin B subunit. Choleratoxin binds to the glycosphingolipid found in lipid rafts, called gangliosides. The nuclei are stained with DAPI. The ventral disc and flagella are also apparent in this image. This image shows a trophozoite that is overexpressing a particular VSP conjugated to an HA tag, stained in red. Again, stained in green are lipid rafts via the fluorescent choleratoxin subunit B. Co-localization between VSPs and lipid rafts can be seen. To analyze microvesicles and exosomal vesicles, they must first be isolated via ultracentrifugation. This is done at 100,000 G for 1 hour and 30 minutes. Vesicles are quantified via the nanocyte instrument. The sample must be loaded into it using a syringe. This is what the vesicles look like through the eyepiece. And this is the vesicles shown through the camera that connects the nanocyte to the computer. When the nanocyte collects data, it determines the concentration measured as particles per milliliter, the size and standard deviation of the vesicle, and the intensity by size of the vesicle. The red values moving across the screen represents the movement of each vesicle being tracked. This graph shows that most of my vesicles have a size of about 200 nanometers, which is the average size for exosomal vesicles. My recent analysis has shown that the most abundant protein found in the vesicles are variant surface proteins, or VSPs, which can link virulence to the exosomal vesicles. My next step is to treat the trophozoites with lipid raft inhibitors while they are secreting vesicles in order to determine if lipid rafts are linked to vesicle production.